Hello everyone. My name is Anna, and I am a bioinformatics software engineer in the Myon Lab here at the Icahn School of Medicine. And today I'll be presenting two related tools for transcription factor regulatory analysis. The first is ChEA-KG, which is a network-based transcription factor enrichment analysis tool. And then I'll show you how we extended this to create ChEA-KG-TS, which is a time series regulatory analysis workflow. So starting off with the basics, transcription factors are proteins which directly and indirectly control gene expression by binding to DNA. And because of this, their dysregulation is often associated with disease. There are 1,639 known or likely human transcription factors, and these form the fundamental units of gene regulatory networks, which are networks of transcription factors and their regulatory relationships. These are also sometimes referred to as transcription factor regulatory networks if you're looking specifically at the transcription factors. And I've included an example of one in the figure here, which is from the sea urchin endomesoderm, to give you an idea of how complex these networks can be. So a variety of computational and experimental methods have been developed to infer and reconstruct these networks. As far as their structure goes, gene regulatory networks have two fundamental units. These are the nodes and the edges. So the nodes are the transcription factors and can sometimes include other genes and proteins. These are the named entities in your network. And then the edges are the regulatory interactions between these entities. So in our case, we're primarily concerned with three edge properties, although edges can include a number of other properties. But we're mostly concerned with the direction, which is assigning the source and the target roles, the sign, which indicates the effect of the regulation, and the weight, which can be used to describe the significance of the interaction. One common way of constructing these gene regulatory networks is a computational technique called transcription factor enrichment analysis. And very broadly, this involves predicting the likely transcription factor regulators for an input gene set based on aggregated libraries of transcription factor target relationships. And these can be collected from a variety of sources, including from literature. They can be from ChIP-seq or ChIP-ChIP experiments, or from positional weight matrices or gene co-expression. So this figure here is summarizing the various sources that are used as evidenced by the transcription factor enrichment analysis tool, Dorothea. There are many of these transcription factor enrichment analysis tools, and I've summarized them here in this table, which is from our upcoming publication in Nucleic Acids Research. But what we've done here is grade the tools based on their inclusions of different aspects that we deem important for a transcription factor enrichment analysis tool. So the key categories are C through F. A and B are just determining whether it's a software tool or a web server application. But C through F show whether each tool has interactive visualization of results, if it provides a network of transcription factors, if the network infers the sign of the transcription factor target regulation, so whether it causes up or down regulation, and then whether the background includes all human transcription factors. And I've highlighted these two tools, ChEA-KG and ChEA-3, which are both tools that were developed in our lab and which I will be continuing to talk about later in this presentation. So the most important takeaway here is that very few of these tools provide both interactive network visualizations with signed interactions using a network that covers all human transcription factors. Another thing to note is if you look at the input, you will notice that most of these tools rely on gene sets or gene lists as input. So this is the primary input that we're going to be concerned with. So there are many ways that you can study and measure gene expression response. And one interesting application is time series analysis. Time series experiments are able to characterize transient cellular responses by taking serial measurements at regularly spaced time intervals. And you can use these to perform RNA-seq, followed by differential expression to produce these time-ordered gene sets. And these gene sets can be produced by doing differential expression between adjacent time points. So looking at hour 1 versus hour 0, hour 3 versus hour 1, and so on. You can look at each time point versus the initial time point or time equals 0, or you can compare them against an independent control group. But ultimately, these gene sets can then be used for input for transcription factor enrichment analysis to identify time point specific regulators. So in fact, many methods have been developed to specifically analyze time-ordered gene expression data. So for example, MOSIG-Pro is a differential expression method, which is tailored to serial data. This tool is aiming to extract genes whose expression is the most time dependent. So the figure on the right here is comparing MOSIG-Pro to other methods. We have MOSIG-Pro only results in row B. Row A is showing MOSIG-Pro and EDGE-R. But ultimately, what you can see is the differentially expressed genes it's identifying are the genes that are changing the most over time. However, MOSIG-Pro does not identify time point specific differentially expressed genes, nor does it identify regulatory networks using these results. So further analysis would be needed in order to do that. And there are some tools that do this. So the DREM suite of tools are based on a method established in the original dynamic regulatory events miner method. And they use this bifurcation method to track gene expression. And then wherever it diverges, they assume this is due to differential regulation because of some regulator. And the way that they are able to infer the regulator is by joining the gene expression with static transcription factor DNA binding data. So there are several other tools based on this method, including iDREM, which shows interactive visualizations, and SDREM, which relates the transcription factor networks to signaling pathways. Two other tools which extend this method are TS Miner and NetSeeker. So NetSeeker is a time series analysis workflow which integrates DREM to identify the regulatory networks. And then TS Miner is another time series regulatory analysis tool that uses a similar underlying method to DREM, this input-output hidden Markov model, but they also integrate pathway information as an additional input. However, these tools are limited in that they require the user to submit DNA protein binding data in order to be able to infer those regulators at each time point. So this can limit the number of transcription factors that are ultimately being considered. And DREM 2.0 actually does address this by providing some static binding data for users to use with their expression data. But as you can see from the table here, the coverage is relatively low. So we only have a few hundred transcription factors compared to the known 1,600 transcription factors that I mentioned earlier. So to address these limitations, we have devised a method to construct a new gene regulatory network which covers all human transcription factors and is based off of our previous transcription factor enrichment analysis tool, ChEA-3. So ChEA-3, as I mentioned, is a transcription factor enrichment analysis tool. The goal is to predict the likely transcription factor regulators for an input gene set. And its enrichment analysis is based on 6 background libraries of transcription factor target relationships. And these are collected from ChIP, ChIP-seq, text mining, and co-expression experiments. Together, these libraries cover 1,632 transcription factors. So this is very full coverage. And this is reflected in this figure from the ChEA-3 publication, which is showing the transcription factor coverage of each of the individual libraries in the background database, as well as the coverage of the composite methods that are used to produce the final transcription factor rankings. And development of this project was led by one of our former members, Alexandra Keenan. So the way that this works in practice is that users can upload their own gene set, and then ChEA-3 will output a ranking of all 1,632 transcription factors in the background database based on their enrichment score. And the reported rank is based on their average rank across all 6 background libraries. This is using the mean rank method. And it means that the higher rank are the transcription factors that are the most similar, whose target sets are the most similar to your input gene set. So the transcription factor rank 1 is sort of the most likely to be regulating your gene set, and so on and so forth. So we had this idea that if we could systematically apply this to thousands of gene sets across a variety of contexts, we could uncover those regulatory interactions, which are consistently observed and therefore most likely to be biologically relevant, and create a full coverage and context-independent gene regulatory network. So this brings me to RMAGEO, which is a relatively new tool from our lab, whose development was led by former lab member Giacomo Marino. But RMAGEO is a search engine that serves almost 180,000 human gene expression signatures that are uniformly processed from studies that have been published to the Gene Expression Omnibus. And this tool gives us access to a wide range of gene sets that are up or down regulated in a variety of contexts. So what we did is we took the subset of RMAGEO gene sets that have clear control and perturbation groups. We systematically submitted them to ChEA-3 and aggregated edges between the top 10 predicted transcription factors and the genes that they were predicted to regulate in each gene set. And then because we know that the genes were up or down regulated within the gene set, we can use this to infer the sign of the interaction as well. So after we built this initial network, of course, we had to filter this down to identify the most significant interactions. So we devised a filtering scheme and did that to produce a final network that has 131,000 signed and directed edges and connects 1,559 transcription factors. So this network was uploaded to a knowledge graph database where it is served by the ChEA-KG web server application. The central feature of ChEA-KG is the network-based enrichment analysis tool, and this identifies the top enriched transcription factors for an input gene set. So this produces these connected subnetworks of enriched transcription factors, which can be used to identify master regulators. So here we have each of these nodes. These are the top 10 transcription factors identified by ChEA-3. And then we have the edges from the ChEA-KG background network. And we can see here that in this context, HMGA1 is involved in several positive feedback loops with other enriched transcription factors, suggesting that it could possibly be a master regulator in this context. So one of the ways that we've extended this feature is to create these ChEA-KG Atlas pages. And these are context-specific collections of transcription factor regulatory networks that are based on specific gene set libraries. So we have 4 of them so far. They're shown here. The first is the cell type atlas, which displays regulatory subnetworks for 131 cell types representing 14 tissues. These are based off gene sets in cell type libraries in Enricher. And then the next one is the cancer atlas, which has subnetworks for 69 subtypes from 10 tumor types from the CPTAC3 cohort. And these gene sets were produced via transcriptomic clustering of each of the tumors and then collecting the upregulated genes for each subtype. And then next we have the mechanism of action, or MOA atlas for short, which has subnetworks for consensus up and down gene sets from 30 mechanisms of action from the Lynx L1000 dataset. And the consensus gene sets were created using signatures from the Lynx L1000 fireworks display tool and grouping them by mechanism of action based on data from the Drug Repurposing Hub. And then finally, we have the Aging Atlas, and this is a collection of regulatory subnetworks for up and down gene sets from 24 tissues using aging signatures from GTEx. And these are all available via the ChEA-KG site, but you can also feel free to use these QR codes as well. So now I'll quickly show you how we have applied ChEA-KG to uncover master regulators of aging and drug response using the Aging Atlas and the MOA Atlas. So as a reminder, the Aging Atlas has gene regulatory networks for 24 tissues. We took all of the transcription factor subnetworks that were enriched for each tissue's upregulated gene sets, and we merged these into a single network. And that's what's shown here. And the node size, as well as the edge thickness, correspond to the number of times that node or edge was observed across all of the tissues. So next, we manually highlighted a few regions in purple here, which show specificity to 1 or 2 tissues. And this shows how combining these networks can reveal some overlaps. So for example, over here, we have a module of transcription factors enriched for both the colon and the stomach. And part of this is overlapping with the module enriched for the brain. Another interesting observation right off the bat is that we can see that the transcription factors ISX, CREB3L3, some of the hepatocyte nuclear factors, including HNF4G and HNF4A, are enriched for multiple tissues. So we had also calculated statistical similarity between between all of the subnetworks using a permutation-based method. So on the left is the resulting heat map of the p-values from that experiment. And we noticed that the tissues in this top module, which includes breast, lung, blood vessel, thyroid, liver, and skin, were all predicted to be regulated by ISX. So this ties into our previous observation. And this is interesting because ISX stands for the intestine-specific transcription factor, and it's typically associated with the intestine. So you're curious how it could be related to all these other tissues as well. So we extracted the composite from the composite network, the subnetwork of ISX and its immediate neighbors, which is shown here. And we can see that it's highly connected to several metabolic transcription factors like MLX-IPL, RFX6, and NR1H4, as well as some more developmental transcription factors like the HNF family. We have CDX1, CDX2, and TBOX10. Ultimately suggesting there is a dual metabolic and developmental role by ISX, which is helping to control how these tissues are evolving as we age. So we adopted a similar global analysis approach for the mechanisms of action atlas, where we started by creating a binary adjacency matrix that just shows all of the edges across all of the networks for all the MOAs. And we found these 5 hotspots of source transcription factors that have similar targets. And these are shown kind of in the middle here. So for each one, we found that the transcription factors in this sort of mini heat map overlap with a specific mechanism of action. So for one, the transcription factors here are from the sodium channel blockers and adrenergic receptor antagonist network, and so on and so forth. And one particularly interesting finding is that for this cluster 4 down here, this overlaps with the PI3K inhibitors. And we can see that there is this tightly connected positive feedback loop involving 5 transcription factors. And actually, 4 of these have previously been found to be associated with the PI3K, AKT, mTOR signaling pathway. However, this zinc finger protein has not been, but because it is highly connected in this subnetwork, suggests that it may be a novel member of the PI3K signaling pathway. So the examples we showed use ChEA-KG to understand transcriptional responses for single static comparisons, but we realized that with the right setup, we could start to tease out the temporal aspects of transcriptional regulation using time series studies. So this brings us to ChEA-KG time series, or ChEA-KG-TS for short. So ChEA-KG-TS is a workflow to investigate time series data sets. And we start with differentially expressed genes at each time point. We can then submit those to ChEA-3 to identify the transcription factors that are controlling the transcriptional response at each time point. And then instead of investigating whether transcription factors at a given time point are regulating each other, we can query the ChEA-KG background network to investigate whether transcription factors in an initial time point are controlling expression of transcription factors in successive time points. And so this gives us more detail into how exactly the transcriptional response is changing over time. So this is a more detailed view of the workflow that we developed. So starting with the time series experiment, first, the workflow automatically computes the up and down regulated genes. You can also upload your own gene sets if you have pre-computed differential expression. And these gene sets, in either case, are then submitted to ChEA-3 to produce the first set of outputs, which are the enriched transcription factors at each time point. Next, we feed those enriched transcription factors to ChEA-KG to create these layered time series subnetworks. So the final output is a UMAP projection of all of the transcription factors in the ChEA-KG background gene regulatory network with the transcription factors that are upregulated for each time point highlighted. And this provides insights into transitions across the different functional modules in the background network. And then the last thing to mention is that development of the original workflow was actually led by one of our summer trainees, Andrew Van Dusen, who's finishing his undergrad at MIT right now. So we applied ChEA-KG-TS to two studies investigating time series responses to drug perturbations as well as exercise. So the first study is investigating the response of triple-negative breast cancer cells to treatment with TRAIL, which is short for tumor necrosis factor-related apoptosis-inducing ligand. But basically, this is a cytokine that has been found to induce the NF-kappa B2 signaling pathway, inadvertently promoting immune suppression in these triple-negative breast cancer cells. So they performed a time series analysis where they measured gene expression in a triple-negative breast cancer cell line at 0, 1, 3, 6, 12, and 24 hours. So we took this data, which was publicly available on Geo, and submitted it to ChEA-KG. And then here are the results. So on the left, we have a time series regulatory subnetwork. So each row here corresponds to the enriched transcription factors for that time point comparison. We're looking at the set of results for each hour versus the initial time point or hour 0. So the blue nodes are the transcription factors that are enriched for the upregulated genes, and then the red nodes are the ones that are enriched for the downregulated genes. And then the green arrows indicate activation edges, and the red edges indicate inhibition edges. But we can see from this that in hour 1 and hour 3, we see several immediate early genes are enriched for both the up and the down genes. And then at later time points, we actually see this activation of NF-kappa B2 and RelB, which are both members of the NF-kappa B2 signaling pathway. And both of these transcription factors are inhibiting the same target. So we have HIVE-P1 and NFAT5. And we can see there's actually a third regulator that is also inhibiting both of these same targets, this ZNF267. So this protein may also be a member of the NF-kappa B2 signaling pathway. And this is actually supported by the UMAPs, which show over here, especially in the hour 12 versus 0 comparison, that ZNF267 is clustering very close to RelB and NF-kappa B2, suggesting shared functionality. Finally, we have a use case from MotorPack. So MotorPack is short for the Molecular Transducers of Physical Activity Consortium, and it is a long-running NIH Common Fund program that studies the effect of physical activity on health and disease. So as shown by the schematic on the right, they collected organ-specific molecular assays from both male and female rats after 1, 2, 4, and 8 weeks of exercise, including RNA-seq data. So the time series RNA-seq from these experiments can be utilized to understand the transcriptional control of specific organs during exercise, and we're going to focus on the liver. So these are the results from uploading that liver time series data set to ChEA-KG-TS. And one thing that we noticed is that in the latest two time points, so after 4 weeks and 8 weeks, again, this is showing the comparisons against our baseline, which in this case is sedentary. But we can see that MLX-IPL is enriched in both of these later time points. And in fact, in the last time point, it is the recipient of numerous incoming positive edges. It's kind of hard to see here, but all of these incoming edges to MLX-IPL are positive. And together, this is indicating that it might be a master regulator in this context. And so in charts B and C, we see that the enrichment for MLX-IPL is mostly coming from the enricher queries libraries from ArchES4 co-expression as well as GTEx co-expression. And if we actually look at these libraries and look at the target sets associated with MLX-IPL, we find that it's been predicted to regulate 3 other known exokines, which are SMOC1, ANG, PTL3, and IGF-1. So ultimately, we have shown that ChEA-KG is a new transcription factor enrichment analysis tool that's able to identify regulatory subnetworks that are predicted to regulate input gene sets. I showed ChEA-KG-TS, which is a new workflow that adapts ChEA-KG to predict time-ordered regulatory networks based on input time series RNA-seq data sets. We applied ChEA-KG to uncover ISX as a possible master regulator of aging and ZNF 367 as a possible novel member of the PI3K pathway. And then we applied ChEA-KG-TS to uncover ZNF 267 as a possible novel member of the NF-kappa B2 signaling pathway and MLX-IPL as a master regulator of exercise response in the liver. And ultimately, future experiments should focus on validating these findings in vitro. So with that, I would like to thank everyone in the Myon Lab and thank you for listening. And especially thank you to Daniel Clark and Andrew Van Dusen, as well as Errol Evangelista, who all contributed and helped a lot with the development of both of these projects. And then these are the QR codes and the links to both of the sites. And just a note to keep an eye out for our upcoming publication, hopefully soon, in nucleic acids research.